Hello, class. On this e-learning day, we'll be learning about the human genome. In order to prepare, you're going to follow the protocol that we always do on e-learning days. So I'll just briefly remind you of that. I want you to have split screen with this recording on one side and a Google Doc open on the other side. You'll be taking notes as if you were in class, which means all of the bolded points on the PowerPoint need to be taken on your notes, and please take all the other notes that you find interesting, as they will help you with the final questions at the end of this presentation. So please get split screen while I turn off my camera and pull up the PowerPoint, and we will begin. Okay, I will give you 5 seconds to make sure you have your split screen with the PowerPoint and your notes ready. 5, 4, 3, 2, 1. We are going to be diving into the human genome. Superhumans, is this a possibility? We think about movies like the Avengers, where we have people turn into superhumans. How about The Flash, that show that aired just a little bit ago? It was one of my favorites. This happened through a scientific explosion. How about a childhood classic like The Incredibles? People who are just beyond humans, superhumans. Is it possible to have superhumans in our lifetime? Superhumans who can go beyond the spans of what we know humans can do. Look at this baby. Look at the feet. You'll notice there's an arrow saying a sprinter. How about the arrow pointing to the ear? Perfect pitch. No baldness. High IQ. Low risk of Alzheimer's, breast cancer, and strokes. How about those eyes? A baby with 20/20 vision. This baby's reaching for some A, T, C, and G blocks. Hopefully, those letters are jogging your memory because those are the building blocks of genes. What this image is showing and foreshadowing is the potential for genetic modification, which eventually could lead to superhumans. Is it possible to create a baby who has all of these different perks and benefits of a high IQ or perfect vision or perfect fitness? That's what we're exploring today. Are superhumans possible? Let's begin with DNA. DNA is your blueprint. As we've learned, deoxyribonucleic acid is found within the nucleus of a cell. There are single chromosomes and pairs of chromosomes, and within those chromosomes, you'll see DNA. DNA has a backbone in the double helix structure. Let's briefly jog your memories on that Google Doc. I want you to list everything you know about DNA structure, and I'm going to give you 30 seconds starting now. Keep typing. Think about the structure. Think about what DNA can do. Think about where it's located. You have 5 more seconds. Hopefully, in those 30 seconds, you were able to think about the basic structure of DNA. DNA has building blocks of nucleotides. So the building blocks of DNA are nucleotides. As you can see on the right, these nucleotides are adenine, guanine, cytosine, and thymine. Those A, G, C, Ts you saw in those blocks are the A, G, C, Ts you see on the right. There are nitrogenous bases of every nucleotide. There's also a phosphate sugar backbone that forms this DNA helix. And there's a hydrogen bond between the bases within DNA. The basic structure of DNA is nucleotides. Those are the basic building blocks that make DNA. So we look at the chemical structure of DNA. As you can see, here are the bases and the sugar phosphates forming that backbone that then forms one side of the strand of DNA. And it's those hydrogen bonds that connect the two nucleotides. Hopefully, this is all review from the last unit that we went over. DNA holds strands together, and from DNA, we get our proteins. So, DNA via transcription gets transcribed into RNA, which is then translated into proteins, and proteins form our bodies. So, when we look at the genetic similarity between humans, a human and a human is 99.9% the same. It is that 0.1% that makes us different, according to the National Human Genome Research Institute. In discovering that 0.1% that's different between human to human, there was the Human Genome Project. As you can see, there are many letters in this photo. Each one of those letters represents a nucleotide base. The human genome is the DNA that codes for genes. This project has the capacity to find the operators, the manual for your body. Within a single cell in your body, DNA would stretch 6 feet. There are 6 billion base pairs of DNA, and it used to cost around $10,000 to code an entire genome. Now it is under $1,000 for this project. The Human Genome Project has increased in efficiency. Have you seen this? Things like ancestry DNA, where you can swab the inside of your mouth and find out your ancestry just based off of your DNA. Your genome's information holds your height, it holds your biological sex, it holds your eye color and your hair color. It holds the potential risk for any future medical problems you may have in your life, susceptibilities towards cancer or diabetic, susceptibility towards strokes. It holds your genetic information and implications for the future. Coming alongside the Human Genome Project has been this incredible genetic modifying ability. CRISPR-Cas9. How does CRISPR-Cas9 work? CRISPR-Cas9 is like genetic scissors. It goes into a human, it's been tested on animals, so it's not proven to work in humans. But following this, there are implications for future work. So CRISPR-Cas9 can go into DNA and it has the ability to edit DNA. So as you can see on the right, CRISPR-Cas9 protein forms a complex with guided RNA in a cell. So CRISPR-Cas9 can specifically attach to a certain part of DNA, and then it can cut that strand. With this comes the ability to potentially put in programmed DNA and insert DNA where the missing strands are. As you can see, that white part that got cut out, new DNA could be put in. This leads to the field of epigenetics, the tags within our genes, the genes that hold and code for all of what makes us what we are genetically. How close are we? There's continual debate on forums about whether or not it's even ethical to consider going from this testing on animal to testing on humans, to go within the genetic makeup of a human being, the A, T, G, Cs, and see if we can modify them. Modifying them has great implications, both the potential for negative effects and the potential for positive effects, because within your genome, you have your vision coded for, you have your muscular buildup coded for, you have your ability to hear coded for. Is it possible for us to get into the genes of an individual, to cut out specific genes and put in new genes? It has been tested with CRISPR-Cas, the ability to change eye color, but can we go a step further into making these superhumans? Heroes like the Avengers or Flash or the Incredibles. This is where scientific research is key. There are websites like .govs or Scientific American or books that tell you more and more about genomics and the potential implications for this research. The point of this PowerPoint was just to open your mind to research that is currently going on and the potential for science in the future. I want you to go through and review with your notes and answer these questions on a Google Doc and submit it to Google Classroom. So you'll just add these questions to the bottom of your note sheet. What is the human genome? What information is in the human genome? What is CRISPR-Cas9? And how could CRISPR-Cas9 potentially be used in the future? I want you to answer these questions and submit them on Google Classroom by midnight on Thursday, May 13th. Also, I would like you to add any questions you have about this PowerPoint on the bottom of your Google Doc, any discussions or reflections, anything you'd like to add, just for fun. But what you really need to do is just answer these 4 questions on the bottom of your note sheet and submit them to Google Classroom. I'm excited to see what conversation this PowerPoint will spark and what different conversations it will spark for our next class time. Thank you so much and have a great day. Take care.